Well, welcome everyone. It's a great pleasure to have so many of you here for the second in our series of webinars looking at the ensuring the reliability of measurements in response to the COVID-19 pandemic. So we have a few very short introductions for you, and then we have three speakers, experts in the field. We'll be able to take some questions, and then we have a discussion session at the end of the meeting. So we do need to finish promptly, so I'll ask everyone to try and keep the time, because I know that a lot of people have other meetings following on. So our intention is to finish at 2 p.m. Paris time here. So just a quick hello to our speakers and panelists. So our speakers today are Professor Maria Zambon, Dr. Corinne Gertz van Kessel, and Dr. Hans Weisers, and they'll have full introductions from our panel members later on. And I'm very happy to have our panelists, mostly from CCQM President Sang-Yul Park, our chairs from the Nucleic Acid, Protein and Cell Analysis Working Group, Jim Huggett, Jeremy Melanson and Jonathan Campbell, as well as Gavin O'Connor from the Protein Analysis Working Group. We're also happy to have Professor Ian Young, who's chair of the Joint Committee for Traceability in Laboratory Medicine, and I have the role of moderator today. So this is a webinar format, not a meeting format. So that means that what we can do is participants can write questions using the question and answer function. Our panel members will pick those up. We expect quite a few questions and be able to ask those selected questions to our speakers as the meeting goes on. For panelists, we're not so many, but if you could use the mute function, switch off your video if you're not presenting or answering questions, and that makes it less confusing for the participants. And finally, we have a last session, which is really an open interview type of session, and then I'd ask all panelists to switch on their videos for that last session. So with that, I'd like to say thank you. That's the structure of our webinar, and we have a brief introduction from our CCQM President, Sang-Yul Park.
Okay, thanks, Robert. Yes, I'm very glad to have the second CCQM webinar on COVID-19 pandemic, especially at this moment. The whole world suffers ever more from the terrible new wave of the pandemic. It is highly likely that there are some serious measurement issues in combating the coronavirus, and we are determined to do whatever we can do as the top measurement specialists and the meteorologists of most countries and economies. We need to know what the most significant measurement problems are in the medical front lines to aim our targets correctly. Several member NMIs have already developed and distributed reference materials for accurate diagnostic testing. Even so, there must be something we can do and we have to do. In this regard, hearing from world-pronounced experts is of a critical value to us. It is impossible for me to sufficiently express my gratitude to today's speakers, Dr. Maria Zambon and Dr. Corinne van Kessel and Dr. Hans Weisers for their precious time and for generously providing us crucial information. With your kind contributions, we'll definitely get more learned and stimulated. We'll listen to the speakers attentively and then share opinions and ideas among the panelists and the audience. Thank you very much again, speakers. I'm also very thankful to our colleagues, Jim and Gavin, who have successfully brought us the excellent speakers today. And of course, thank you very much to Robert and the BIPM staff who must have worked hard to prepare for this online seminar. We also have a very respectable guest today, Dr. Ian Young, the chair of JCTLM. He is also an expert about this COVID-19 pandemic and the highest ranked consultant to the UK government. Thank you very much, Ian. And many thanks to the other panelists, Jeremy, Bunt, and Jonathan. And all participants, my CCQM colleagues, I hope you all to have a very exciting and fruitful time throughout the webinar. We are doing our works as usual, but at this particular time, we ever more feel that we work for humanity. Thanks a lot to you all. Robert?
Thank you very much for the introduction. So, Maria, could I ask you to share your presentation? And Jim, could I ask you to introduce Professor Zambon, please?
Certainly. Good
Hopefully,
day, everybody.
you can see something there.
Yeah, we can see the screen. Thank you, Maria. I would just like to second the President's comments in thanking our speakers today.
Jim, could you speak up a bit? Sorry, can't hear you.
I'll shout. I would like to thank the President's comments and thanking our speakers today. Our first speaker is Professor Maria Zambon, who is Head of Influenza and Respiratory Virology at National Infection Service at Public Health England. And it's not too big a statement to say that Maria has pretty much been one of the most instrumental individuals in the response of the UK to COVID-19. And we are really, we're really lucky that Maria has been able to provide us with the time to present her experiences. Maria is also the director of the WHO National Influenza Centre, the WHO Coronavirus Reference Laboratory, and the RSV Reference Laboratory. So without further ado, I'm really looking forward to hearing your presentation, Maria, and over to you. Thank you.
Thanks very much. Well, hopefully you can see my screen, so let me know if there is a problem. So the themes of today's talk really will be about development of diagnostics in an emergency, how you make sure that they are accurate diagnostics, and some of the issues that crop up along the way. And in particular, in discussion with Jim prior to the sort of presentation, he asked me particularly to sort of identify areas that standardization or provision of additional materials could be helpful for all of you to think about, and some of the lessons learned through this process. So first off, let me say a few words about coronaviruses. These are the largest RNA viruses, and they're called because of their morphology, because they have, in the electron micrograph shown on the left of the screen, you can see that they have a ring of sort of spike proteins, which gives them the term, corona. And at the bottom, you can see a linearized genome, which sort of sets out the size and organization of the genome structure of the coronavirus, which are enveloped RNA viruses. Now, when in recent years, reverse transcription, PCR, has taken the key role in identification of viruses in humans, compared with 20 or 30 years ago, where we relied on the ability to be able to culture virus. And what's shown here is very quick schematic of a virus getting into a cell, replicating and coming out. And in the case of a coronavirus, this is cytoplasmic replication. Now, the point of this slide is to say that RT-PCR can be used to detect the presence of specific genetic material in clinical samples, so viral genetic material, which can be either virion, what's in the virion, or what's in infected cells. And there are many different macromolecular replicated intermediates in infected cells. But PCR itself cannot distinguish between infectious and non-infectious virus, which is something to keep in mind as we go through. Now, also, when you are thinking about different virus detections, whatever they happen to be, you also need to think about the diversity of viruses within the same kind of genetic groupings. And here, when we're talking about coronaviruses, we're well aware of the fact that there are a diverse group of coronaviruses which infect humans that we already know about. And we also know that the potential for this biological grouping to deliver emerging viruses is already proven. So SARS-1 in 2003 emerging from mammalian, small mammals into humans, or bats into small mammals into humans, then MERS coronavirus 2012, and now SARS-CoV-2 coronavirus in 2019. So one needs to think, when developing diagnostics, to be aware of the diversity, but also the similarity with other viruses. So in relation to the development of diagnostics for SARS-CoV-2, once it was clear, the very early signals right at the beginning of January, that there was a problem with a new coronavirus, WHO did what it often does in these circumstances, is bring together teams of expert laboratories, or laboratories with appropriate expertise, but also facilities to do the development work. And that, in this case, actually delivered the first sort of international development of diagnostics for coronavirus, now which was published towards the end of January. The reason I highlight this is because amongst the team of authors there are the assembly of people who had worked together for SARS-1 in 2003, and then many of those people also working on MERS during the emergence of MERS in 2012-13. So an expert group, but not only expert in knowing about virus biology, but expert in having reagents to hand. And that's absolutely critical when you are dealing with things in emergencies. You can't wait for stuff. You need to have what you've got in the fridges and freezers to do things really quickly. And shown above is the linearized genome, which shows the key target areas that were selected, providing the potential for confirmatory strategies, which I'll talk about in a minute. So when you're developing diagnostics for a new virus, you do tend to use the experience you already have, and you use several principles. Usually design several different assay targets so that you have the ability to confirm different things. You have increased assurance if you've got more than one target positive. In the case of coronaviruses, what I hope you can see on this screen is that the schematic here of the virus genome is accompanied by I think it's a northern blot here that we've got of viral RNA fragments. And what you can see on the polyacrylamide gel, with decreasing molecular weight of RNA species, increase in density. And what that does is it tells you that there is a transcriptional gradient across coronaviruses, meaning that virus replicative intermediates are increased by 100 or 1,000-fold towards the 3' end of the genome, which means that if you are developing diagnostics and you're not sure whether you're going to be detecting the virion itself, which has a single copy of RNA, or replicative intermediates in an infected cell, you want to try and target your diagnostics where you think you will have most target to find. And that's one of the reasons why the diagnostics that are selected by the WHO group, which I showed on the previous slide here, are primarily targeted towards the 3' end and centrally at the RDRP, which is the coronavirus polymerase and the most highly conserved region of a coronavirus. So looking to try and give the most assurance for detection, which also deals with cross-specificity. So when you are looking and designing targets, you do need to make sure that you have the ability to eliminate other seasonal coronaviruses. So the materials needed to develop diagnostics are obviously knowledge of the viral genome. So the absolute sequence is really important, and we were given a kickstart there from China on the 10th of January with the publication internationally of the virus genome from Wuhan. But between the 1st and the 10th of January, there was already development work underway based on SARS-1. You need to have virus template and use either virus infected cells, virus particles, recombinants, transcribed RNA, whatever material is to hand to provide template, and ideally panels of clinical material which are well validated so that you have knowledge of the clinical case definition that has given rise to that material. So the type of materials you need for standardization at that point would benefit from having rapid full-length RNA transcripts, which we did not have, and we had to rely on short fragment RNA transcripts, which were made in one particular laboratory and then distributed to the actual group who work together to actually develop the PCRs, and you ideally want quantitative material. So when you're doing things in a hurry, and you may need to rely on viruses that you've got or virus-related material, and there are then issues about how do you quantitate virus, because there are many ways of quantitating virus, and they don't all tell you the same thing. A multi-cycle replication, as shown in the top right-hand corner, will give you information about plaque-forming units or individual infectious particles, but is dependent on multi-cycle replication and the ability of that virus to spread cell to cell. Whereas a focus-forming unit, which is a much quicker way of quantitating virus, may tell you about viruses getting into cells, but not necessarily viruses able to spread. So you could easily find a 10-fold difference between multi-cycle replication, quantitation, and focus-forming units. And then if, for example, you went for an EM particle counting approach, you could easily find 100 or 1,000-fold difference between the number of particles that you can see and the ability of a virus to spread through an infectious biological assay system. So the reason I'm highlighting this is that methods of quantitation of virus to develop standards may also themselves be quite variable, but it is a benefit to think about different ways of doing this and to use multiple ways of quantitation when you're developing diagnostics. So then moving on, assuming you've got some template, whether it be live virus and you've decided which method you're going to use, or synthetic template, it's necessary to do a probit analysis whereby you have multiple replicates of dilution series to try and give you an endpoint detection analysis of copies per reaction, which is a way of providing some kind of assurance of what it is that you're detecting and what the limit of detection might actually be from an analytical perspective. This is the work that we did, but we didn't at the time have SARS-2 virus, and we had to make do with the use of SARS-1 virus. So again, highlighting when you're developing diagnostics in a hurry, you need facilities because this is a select agent in many countries. It requires highly restricted facilities and very experienced staff to actually work with the virus, or alternatively to use short copy RNA, whatever was available, synthesized very rapidly in one laboratory to match the template of the actual sequenced virus, which was then shared again very rapidly with a small group of laboratories who shared data and approaches to try and work out which were the most useful assays. So what was published was a fraction of what was actually looked at to make sure that there were adequately performing assays. And so when you finish with a kind of description of analytical performance, you're usually looking at a whole range of things, that is sensitivity and specificity against panels of clinical material which are from appropriate syndromes taken at various time points post-illness onset. And in the case of the work that was done here, I would say that over the course of a few days, we actually pulled out of the freezers up to 1,000 different samples appropriately organized for knowledge of cases of respiratory infection, or diagnosed with other known respiratory viruses to allow sensitivity and specificity estimates to be made. And what I've highlighted in red is the fact that one also needs to think about disease prevalence and the positive and negative predictive value of the assays that have been developed, which I will come on to later. So part of the analytical performance is actually understanding how sensitivity and specificity might differ according to the disease prevalence. So assuring the quality of diagnosis does mean some form of clinically validated assay using validation panels and an assessment of sensitivity and specificity. But associated with that needs to be well-trained staff, management and facilities, as I've just indicated. So the development of diagnostics in an expert laboratory is one thing. Another is providing diagnostic test results, potentially disseminated out to a much wider, different skill mix environment. It helps once you have the right kind of material. So our first case was detected on the 31st of January, or 30th of January as it happens. We used multiple assays, different targets, eliminating for sure the seasonal coronaviruses with concurrent analysis and sequence confirmation. What's shown in the box here are W1, 2 and 3, which were the new specific assays, which we ran throughout January on every single potential case that came to light in the United Kingdom, along with a seasonal coronavirus assay for 4 human seasonal coronaviruses, and also a pan-coronavirus assay, which we knew would pick up every single known coronavirus, but different format, block-based as opposed to real-time, which gave us very good assurance of that first case, or first 2 cases, in order to be sure of what we actually had. And as soon as we had new cases ourselves, we could look into the proteomic analysis of the SARS-CoV-2 and make sure that it actually performed as we expected it to from the perspective of viral transcription and production of viral macromolecular products during replication. In other words, verify that it did indeed act as a regular coronavirus, such that we were targeting our diagnostics in the right place. So the early clinical service delivery in the United Kingdom changed very rapidly. There's rapid evolution of tests, changing laboratory algorithms, different requirements for standards and controls during this early time, and obviously the rapid necessity to disseminate capability, which at the time required that we provided control material for the development and dissemination of testing capability. And the very first control material we provided was SARS-1, which we knew would work in the assays. And then we also provided, as best we could very quickly, quantitated new virus, which was provided from our virus isolation from that first case, because the things that we might have liked to have had at that time were not available. So in disseminating capability, we had to provide all of the materials that went with it. So the challenges in those very early days were the quality assurance of reagents, in particular commercially supplied reagents, primers and probes, the supply of reagents as companies were struggling to ramp up to meet the enormous global demand, the actual sampling methods, which I'll say something more about, and the responses to queries and the guidance and support needed. In order to disseminate within country, it was necessary to provide detailed protocols and standard operating procedures, control materials, as I've just mentioned, and to set out a methodology for equipping laboratories to provide assured results, which was ensuring that the assays and the protocols that were provided did actually perform as expected against panels of materials in their own laboratories, which would have been negative, that is pre-pandemic materials, but also performed as expected against dilution series of materials that we provided with probit analysis and against different controls, and then the final assessment the distribution of blinded panels of spiked clinical materials, which we asked laboratories to evaluate in a timed assessment. So building capacity early in the early days was, first of all, in February, distribute the protocols and procedures to public health laboratory system, and then further out to the clinical laboratory systems. And as government developed capacity for mass testing into our mass testing laboratory networks, so 3 different networks that are involved in delivery of capacity, which at the very earliest only involved the public health system and the clinical laboratories. So the clinical performance could best be described as sailing the boat while you are building it. One has to think about what do you know in the face of a new virus. So SARS-1, which is genetically very closely related, was a starting point for thinking about what this new virus might look like. And based on SARS-1, we might expect that virus would be found in a range of different biological samples and over a different period of time, potentially quite a prolonged period of time, and that we might see illness and recovery, peaks of virus shedding at different times post-illness onset. So early information about timing of illness to incubation came out from the Chinese experience, which suggested, shown in the top blue panel, that the incubation period from exposure to illness onset was consistent with what had been seen for SARS-1 and also MERS virus at around about 5 days. What was not known was the relationship between symptomatic and asymptomatic infection, and that continues to be something that is problematic for understanding disease transmission. But working out whether you have asymptomatic infection is critical for public health control measures. So then, as we came to look at our first 9 cases, we were clear that we needed to sample in multiple different biological compartments to check to see where virus was actually being detected. So the black here, each panel here, A to I, represents an individual who was sampled sequentially from the day of the day in which they came to light, through different body compartments. So black represents detection, grey represents sample but no detection. So one of the things to highlight here, in particular in case I, is that we see in the upper respiratory tract, which is what URT stands for, black, grey, meaning that you have already the information that when you are sampling, you may well find people who are positive, negative, positive, negative. So a sort of variation in detection capability, and a small, in this instance, I think it was 2 out of 8 or 2 out of 9 who had detection of virus in fecal material. So showing us that we may need to look in more than one body compartment. Again, with development of diagnostics, it's important to think about sample types. Which area of the body are you sampling, and what kind of samples are you taking? One of the things we found from our early analysis was that lower respiratory tract samples gave us higher viral loads as measured by lower CT values, but sputum is not a particularly easy sample to get. Most cases did not have lower respiratory tract secretion, so upper respiratory tract materials were going to be the ones that we needed to look at. And then when we looked at nose versus throat samples, we didn't find much difference between nose and throat sample detection of virus, and we went with a combined nose and throat swab sample as being our optimum sample for detection. We also looked in certain clinical outbreak settings at detection through oral fluids, and found that for us, a nasal swab was preferable for detection rather than oral fluid. And so within the UK, we have standardised on use of nose swabs. As we developed data, we saw that the kinetics of RNA detection really peaked towards the beginning of illness, and we could see that as illness developed, there was a decrease in detection of amount of virus over time. But we also saw pretty early on that we could continue to detect virus for quite some time, well up to 30 days and beyond. And that duration of detection has since been borne out in systematic review, with the maximum duration of shedding being at least 80 to 90 days, which is a recent systematic review summary. So that tells that one is going to be detecting by PCR for quite some time. Again, this business of using different sample types is quite important because it makes a difference in time to undetectable, but time to undetectable using different samples, that is upper or lower respiratory tract samples, may not be relevant in terms of infection control, which is what you're particularly concerned about. So the fact that you can continue to detect virus in a more restricted body site doesn't necessarily correlate with infectiousness. We were keen to look at CT values in symptomatic and asymptomatic individuals as they came to light. And we also did virus isolation from these various different groupings, because it is quite important from a public health perspective to work out who and where are the transmissions happening from. And in this piece of work that we did based on care homes around about April, May time, that was really the first insight that we had, that asymptomatic people were as infectious and had as much virus as those who were symptomatic. And that was also borne out in the kind of culture-based work that we did, where we used the ability to cultivate virus as a proxy for infectiousness. And in particular, through this work and other clinical studies that we did, we found that there was a correlation with times from symptom onset to the ability to cultivate virus. And when we analysed all of this, by about day 10, pulling it all together, after about day 10 post-symptom onset, there is about a 5% chance of being able to recover infectious virus, which is a proxy for infectiousness. And on the left-hand side here is the data that we took from around about 250 cases where we had virus material or clinical material coming direct to the laboratory. So a really good chance of being able to isolate virus if it's there, showing very nicely the relationship between CT value and infectious virus recovery. But plainly, ability to cultivate virus is not something that all laboratories can do. And we need to think about developing other ways to provide surrogates for infectious virus detection. And that would include potentially the use of subgenomic RNA, but quite a difficult one to think about for control material, and perhaps we'll come on to that shortly. So that then allows building of what do we think is the kind of natural history of infection and the time at which maximum infectiousness happens, which is likely to be early from symptom onset. So next up is thinking about how to scale up. Often this is a political imperative rather than a scientific one, and it was felt that in the UK it was necessary to have massive mass testing capability from early on. And this is where thinking about the predictive value of your tests is really important, because positive predictive value does tell you something about how much you can rely on a positive result or a negative result. And this, I think, is kind of analysis is something that's very common for people who think about mass screening, that when the population prevalence becomes low, additional steps are needed to prevent too many false positives coming to light. So what's shown in the graph here are 3 different test specificities, which show the difference at low prevalence of population prevalence. And the point being that if you have a test which is only 98% specific at very low population prevalence, all of the positives that you get are more likely to be false positives than true positives. So you do need to have confirmatory strategies. There is then necessity to think about operational performance when you're using more than one target in your assays, and quite often one useful technical tip is simply repeat the analysis if you end up with a small proportion of things that you cannot quite sort out. So this is an example of, I think, 3 weeks worth of testing early November here at Colindale, where we ended up with 1.4% of things that needed repeating, some of these being due to single target test only, as in a duplex test, where you actually have to try and work out what are you actually dealing with. And repeat analysis does often give you resolution, giving you overall a very low proportion of indeterminates, which is certainly what you want operationally. But analytical false negatives can arise because you've got movement of viral genome, or because you've got unusual sample types which contain inhibitors. And the quality of specimen is therefore extremely important. So one way of looking at that is how much human cellular material have you got present. And if you have inadequate sampling, you can see from analysis of these figures, which I won't go into, your ability to detect a true positive is reduced significantly if you don't actually have enough material. So thinking about the quality of the specimen itself is particularly important. I perhaps won't spend time on that. False negatives is something that is also extremely important. There's a very nice New England Journal of Medicine article that drew attention to this, and this shows quite nicely the difference between a low sensitivity test and a higher sensitivity test, which relates to pre-test probability. So actual clinical screening itself is quite important, and that itself is relevant when you're thinking about changing case definitions, which do change in an emerging situation. So the turquoise spikes here are where we set out to sample serologically around about 250 people who were test negative very early on to see whether or not we were missing things through PCR as a result of the type of sampling. And all of those, I'm pleased to say, were seronegative, which is not something that we could actually do now. So false positives are again another problem, and I think I've mentioned here the issue of a long tail of detection in somebody who has had infection. So the 2 dotted areas represent times when the CT values will be low. CT values will be high, but the amount of virus is low, and there will be different clinical implications. Early in infection, virus first becomes detectable, but that person is likely to be highly infectious, whereas towards the end of infection, that person will no longer very likely be infectious. One of the problems with emerging virus disease is great diversity in methods that are being used. So here we are. These are the clinical laboratories in August in the United Kingdom, with a huge diversity of methods. In November 2020, quality assurance schemes had been developed, and showed that over 95% of tests being done in a wide range of laboratories were still targeting those targets that we had picked out in those early days, but again showing diversity of target approaches. And here was an FDA analysis which showed very nicely the comparative performance of some of these diagnostic tests as measured against a single reference standard, which is exactly what you need to try and work out issues about sensitivity. So a lot of interest in the use of saliva, which is highlighted in red, whereas at the bottom are the most sensitive tests and at the top the least sensitive. Evolution of testing over this period of time has incorporated now going to point of care tests, again, multiple different platforms, multiple different technologies, including starting to be combination with other respiratory viruses. So let me just sort of finish up in the last couple of slides with talking about the phases of assurance. So what's shown in the top here is a sort of schematic of first wave and then moving into second wave. So as we start with a new virus, the first thing that's important is development of capacity, which is then supported by the necessity for a good reagent supply chain. And this was a critical problem very early on. Community testing schemes, as expansion of testing takes place, led to supply chain shortages for swab types and the use of absolutely every swab type under the sun, including genital swabs, for sampling respiratory tract. Concern at this point is about false negatives. What are we missing? Because we know that disease prevalence and incidence is on the rise. How do we contain the spread of a new infection? As we go on, we go through the first wave, we're coming into the second wave, or at a period of low prevalence. The concern then is about the false positives and the implication that those cause. As we develop capability, there is increasing complexity of testing, and then the introduction of mass testing using less sensitive methods to try and adopt different strategies for identification of disease with the evolution of testing types. And so for each one of these stages, the different kinds of quality assurance things that are needed is different, and there are multiple gaps. So for future planning, there are definitely things that we need to think about that will improve the assurance as we need to expand, should we be faced with this kind of situation again. And I'm pleased to sort of finish up by saying that one of my points here is the amount of time it takes to do things. Normally it takes 2 to 3 years to establish an international standard, but I understand that possibly even today or tomorrow, WHO will propose or endorse the establishment of the first international standard for SARS-CoV-2 RNA, which is in fact, I believe, going to be proposed to be that very first England isolate that I told you about earlier on, assigned an arbitrary unitage, which can be used to help standardize detection strategies. So my conclusions here are pretty obvious. Emergency response diagnostics is very challenging. Testing strategies may be politically rather than scientifically determined, and therefore one needs to respond flexibly, agilely, and with innovation. There is a need for more rapid response at every stage for provision of standards and reagents, and quality assurance support for industry as they develop capability needs to be seriously enhanced. So let me stop there. I hope I'm not too long over time, and provide the acknowledgements of many of my colleagues here in Public Health England, in our regional laboratories, and the WHO Reference Laboratory Network. Thanks, and over to you.
Thank you very much, Maria. That was
Can't hear you very well, Jim.
Yeah, I'm sorry. There's something wrong with my thing. Can you hear me okay if I make my
Yeah,
voice?
no, that's fine. Yeah.
Thank you. That was amazing. Absolutely brilliant. Thank you so much. And Neil Almond has stated on the chat that actually it's been the WHO material has been discussed in Geneva as we speak. I'm going to move on now. We have a question or two, but I think they'll be better to have them at the end. Because they will also potentially be relevant to the following speaker. So thank you very much, Maria. We'll speak to you in a second. That was wonderful. And I'd now like to invite Dr. Geertz van Kessel to share her screen. And while you're doing that, we're very grateful to have Dr. Geertz van Kessel, who's going to be talking to us about the rapid antigen testing, which certainly is becoming more commonly discussed in the UK, and I'm sure that's the case globally. Dr. Geertz van Kessel is a clinical virologist at the Department of Virus Sciences in Erasmus in the Netherlands, and I welcome you, Dr. van Kessel, and thank you very much for your time.
Can't hear, you're muted.
Okay. I think you can hear me now, right?
Yep, that's great. Thank you.
Yeah, I just wanted to check that you can see my slides as well, but I assume that's fine. So I'm Corina Geertz van Kessel. I'm working as a clinical virologist at the Virus Science Lab at Erasmus MC. I would like to thank Maria for the very nice talk, which introduces a lot of topics which I will address as well. In my talk, I will specifically focus on the challenges in validation and implementation of rapid testing against SARS coronavirus. If we look into the news and papers and all kind of media, we can see that there's a lot of attention for rapid antigen testing, and it seems to be kind of a promising tool to stop the pandemic and to help us a lot. And I think it's very important to think about these assays and how we can use them and how they can be implemented and in which amount they can help us. So when we look into the standardly used rapid antigen tests up to present, these are manual tests based on a chromatographic immunoassay principle, of which the target is mostly the nucleocapsid protein. So the assays are coated with antibodies which will detect the nucleocapsid of the virus. The assays are of course quite inexpensive related to molecular tools. They're quite easy to manufacture, they're easy to use, easy to handle. They will lead to a fast result. Generally in approximately 15 minutes you will have the result, which will result in, of course, it can lead to rapid contact tracing if you would implement these tests. But the question, of course, is what about the performance? And a known drawback is the decreased sensitivity of the assays. But how much of a problem is this decreased sensitivity once we would like to implement these assays? Well, when we look into this slide showing the kinetics of SARS coronavirus infection and the diagnostics needed, we can see that, and we know very well by now, that viral RNA can be detected early upon symptoms and sometimes even prior to symptoms. And the detection will often last long after the clinical symptoms have resolved, which is being depicted in the gray plot. The question which we need to answer is how to detect infectious individuals. At least that's the question for the public health issues we're facing. And to answer that question, of course, we need to know when exactly are individuals infectious. And we have ideas about that, but it's not a clear cut. We have to work with the range among which we think that people are able to transmit the virus, which is the pink box in this plot. The idea of antigen testing could be that although it's less sensitive than PCR testing, it might very well correspond to this infectious period. And the thought is that it likely will be, but it's also important to know how much will you miss specifically in this early phase, because this is the time where people come for testing in the community. So what we want to answer is how exactly can we determine the analytical sensitivity of the test? And that will be the first question when looking into these assays. But other questions and recommendations which have been done by the WHO are that prior to thinking about this analytical performance and the performance which you require, you need to think about what will be the timing of the sampling in the patients you will test. Also, what will be the population in which you want to implement the test, and the expected prevalence of SARS coronavirus in this population or in time. At least you have to take it into account. As Maria showed already, this will really affect the predictive values of your assay. Another thing you need to take into account is the sample collection. Will you work with self-sampling? Will there be professional sampling? And what will be the right choices here? Finally, there has been discussion that the severity of disease should be taken into account when validating these kind of assays. But by now we know that severity of disease does not really affect, the amount of virus detected is not affected by the severity of disease. So the question is if we should really validate these assays in different severity, because in this case, we want to know if the assay detects the virus which you're interested in, so the infectious virus. First of all, the analytical performance of antigen test versus PCR reference assays, so versus molecular reference assays. The big problem which we faced there up to present, I should say, is that up to now, this international reference standard was lacking, and it made it quite hard to adequately compare different antigen tests with each other, but also to compare results from different laboratories. So it's very good to hear that at this moment the discussions are ongoing, and we will probably soon have access to this standard, because it will make some things easier, I think. So what laboratories are now doing is they're using genome copy numbers to make a comparison. Of course, there are a lot of laboratory standards which have been used in PCR testing, which are now also being applied to the antigen testing to make a comparison between assays. Quantified virus stocks can be used, and of course, quantified recombinant proteins are useful to compare the assays and to say something about this analytical performance. But still, we need to answer the question, what is the viral load which is crucial to detect, and under which viral load can we consider a patient as not infectious? Well, to look into infectivity, like was already introduced, we also have used cell cultures as a measure of infectivity. What we did is we collected respiratory samples. These were directly transported to the lab and directly put on vero cells, as we can see on the left side. Quite soon after infection of these cells, you can see a cytopathic effect on the right side. The cytopathic effect is always confirmed by specific staining. So these cell cultures have learned us in a set of hospitalized patients that the median viral load was significantly higher in culture positive samples than in culture negative samples. And that's exactly what Maria showed as well in a population from England. But we should realize that this is a hospitalized population of patients, and that it might be different in other populations. So in this plot, I show on the left, on the y-axis, the RNA copies per ml. The duration of symptoms is on the x-axis, and what we see in black are the culture positive samples. So we see that there's a clear cloud in the left top of people with infectious virus. When we put this in a probit curve, we see that the probability of isolating infectious virus becomes less than 5% when the viral load is below 6.6 log RNA copies per ml. And this quite well correlates to a paper earlier this year by Wolfel et al., who shows that the cutoff is approximately at 6.5 log RNA copies. So I mean, this showed that that these results are quite comparable, although the paper of Wolfel had a bit of a larger range as they included less materials in their paper. Nevertheless, as I already started, it's all about the population you're looking in. These are hospitalized patients, and what we know is that they are often at least a week after the start of symptoms. And this is, of course, different once we test in the community. In the community testing, we mostly test mild symptomatic people who come very early after the onset of disease. So here we see that the majority of people which came to our community testing stations came prior to 10 days. The majority also had quite high viral loads. And when we look into the cultures of these viruses, and we put it again in a probit curve, we see that the community samples are plotted in red. The paper from Wolfel earlier this year is in green, and our hospitalized patients are in blue. So we see that the virus culture in these community individuals tested with mild symptoms, they had the virus culture became positive at a much lower viral load. So what does this exactly mean, and how should we take this along when thinking about testing? Well, it's important to always realize that once you're looking into different populations, they will be in another phase of disease probably, and they will have another immunological status. So what we looked into in our hospitalized patients is the amount of immunity that they had at the moment of doing the viral cultures. So we measured this immunity by virus neutralization with a PRNT test. So this is a plaque reduction assay, and it reflects the amount of neutralizing antibodies somebody has. And we see that once the titer of these neutralizing antibody increases, the number of positive cultures dramatically decreases. And actually there was quite a nice cutoff to put here. So above the titer of 1 to 80, we did not find any positive cultures anymore. It's important to realize that this virus neutralization assay, of course, as well is dependent on the laboratory and needs an international standard as well to really set an international standard of a titer for these kind of assays. And that's something that we are currently working on. But it's important to see that in these hospitalized patients where the ability to culture the virus was less than in the community patients, it's probably due to this immune responses which they have already mounted, whereas the people early in disease do not have them. This again is a probit curve where we show that this probability of isolating infectious virus is less than 5% once this PRNT is at least 1 to 80, and we show it here. So then let's go back to the ability to culture virus in the community, because this community testing is actually asking for rapid testing, and antigen tests can really be a solution there because it will allow rapid contact tracing once this works. So again, here I show the probability to culture the virus with the different PCR viral loads. And in the bars, I now showed the amount of samples which were culture positive. And I used this plot to depict the rapid tests which we tested. And these were 5 rapid tests which we compared. And what you can see here is that there's quite some difference in the cutoff of the tests, in which they still were able to detect an individual as positive. So we see that these tests by Abbott and SDBio actually resulted in the best performance. They had the lowest level of detection, and that's why we chose these assays to look further into their performance and their utility in testing larger populations. We see, and the fact that we see so much difference in these antigen tests really, again, highlights the need of an adequate comparison and well, preferably with a standard which can be used to compare these different assays with each other. Here we set up the numbers of these different assays. So A were the two best assays which I just showed, and when calculating their sensitivity in the mild outpatient population, it was quite good, 97%. And we also calculated what it would mean with the probability to culture the virus in the other population. So in the mild hospitalized patients from the paper of Wolfe et al, and from the severe hospitalized patients, which we described earlier this year. Yeah. So what I just would like to say is that it's the two tests which we think are most useful are the test by Abbott and by STB. So the next point is the sample collection. There's quite an urge to use the antigen rapid testing by self-sampling. And although that could, of course, be a very good choice, I think we really need to think about the kind of samples which then are useful for this kind of testing. Of course, we've heard about testing saliva. We can test nose or throat swabs, oral fluids. There's all kinds of possibilities. We know that there's not too much difference between throat swabs and nasal swabs. So these are the most likely to use for these antigen devices. But I think what is important to realize is that as already also shown by Maria in SARS coronavirus, there's quite some difference, and people can be positive one day and negative the other day. And that's something what we know very well as well from other studies. And in this study, we show a similar trend for example, rhinovirus detection. It's a study done on the ICU some time ago, but it very well depicts that even with professional swabbing at an intensive care unit, when comparing a nasopharynx swab with a tracheal aspirate, there is a huge difference in detection and a very large variability. So as we can see here in the lower plot, in blue we see the nasopharynx swab, and in red the tracheal aspirate. And we see that although the tracheal aspirate has some drops as well, the nasopharynx swab, well, shows a lot of variation. And I think it's something to take into account when thinking about self-swabbing, because the variation will become even larger there. Another question which is mounting is ideally antigen tests would need very frequent testing. It should be very easy accessible, and perhaps people should be able to do these tests themselves. So not only the self-swabbing, but also the interpretation of the assays. Well, as we all know, probably the assays are read by a line. And once the assay is clearly negative, we only see a red line at the control point. And usually the negative tests are quite clear. The very high, the patients with very high viral loads have a very clear result as well, and are probably very easy to read. But the problem comes right at the samples where there is, where we're at the cutoff of being the detection limit of the assays, where we can see that it can become quite difficult to reliably read these tests. And that puts some questions in how easy we can rely on well, reading the tests at home or in a non-professional setting. Yeah, I think it's good to realize that there are automated systems coming on the market now to do bulk antigen testing in a laboratory setting, of course, but that might offer an alternative for these kind of manual tests. Finally, I would like to go to the implementation and which kind of settings would be of interest there. Well, the settings which are often discussed are settings in which we know that there's a high risk. Of course, there are the high risk healthcare environments where you would like to reduce outbreaks. There are schools where you would like to do rapid contact tracing and also reduce the amount of outbreaks, student campuses, but also the larger events who are really eager to start up again. Well, there are questions about the use of these antigen tests in these population. As I already stated several times, this test sensitivity of the assays can be increased by testing frequently. And thereby the positive thing is that once you test very frequently and you have a positive case, you can rapidly start up the contact tracing. But still, we should realize that the test sensitivity, so the analytical sensitivity, should be good enough to allow these kind of implementations. So what we did to bring the testing towards the population is we used several mobile labs. These mobile labs can be placed in areas where there's a high level of circulation to allow people to easily test themselves, and also to allow asymptomatic testing by these antigen assays. And this has worked quite well. So it was really, people were coming easier, and there was less of, so we have some areas in Rotterdam, for example, where there's a lot of people with a low economic living standard. They do not always have the availability to go to the large test centers. And it really helped to do this kind of testing and to allow rapid results and a rapid turnover as well. So we saw some benefits there. Another question is, can we use the antigen tests to do the massive testing, as Maria also introduced, which is often a political choice, to test complete cities, complete areas of the country, talking about 10,000 to 50,000 to 100,000. These are huge amounts. In the Netherlands, there's kind of a pressure to use antigen testing in this kind of settings. Well, here we see one of the technicians performing the manual antigen tests. And I think we can all imagine that these manual tests do not seem very appropriate for this kind of use. But as I said, there might be automated systems which can help these kind of settings to increase the amount of testing and the turnover of the testing. Finally, I would like to end with a paper from colleagues here in the Netherlands who commented on the urge for implementing rapid antigen testing as the solution for reopening the society. They did a mathematical modeling study, and they showed that once the reproductive number is still at 2.5 and all the control measures would be lifted, it would require 100% of the population being tested every 3 days with a rapid test, with a similar sensitivity as the PCR-based tests, to really be able to allow this. Well, I think we all agree that that is not a realistic setting. So I think that the political pressure on antigen tests and seeing it as a solution for everything, I think we should be careful with that. But that, of course, the assays can be a very useful tool in specific risk settings. I would like to conclude that I'm very happy to hear that the universal standard to compare analytical performances of, for example, the antigen tests is coming, because it's very much needed. I showed that a virus culture system could very well serve as a measure for infectivity and can thereby support the analysis and the comparison of these antigen tests, specifically because these antigen tests will focus on infectious individuals. So it's very important to have a surrogate marker there. And finally, I would want to say that it's really crucial to identify the population and the intended use of the rapid antigen tests prior to evaluating, and of course, prior to implementing the assays as well. I would like to thank you for your attention, and these are my colleagues with whom we have been working on the analysis of the rapid tests.
Thank you very much. Again, really great talk. Really appreciate that. Some really good details there. I'm going to propose we move on, though, and save the questions, of which there are a few, to the end of the session, if that's okay with everybody. I will hand over to Dr. Gavin O'Connor, who's will be handling the rest of the presentations. Thank you.
Great. Hopefully everybody can hear me. Hans, I suggest you load up your talk while I introduce you. As the final speaker in our session, it is our pleasure to introduce Dr. Hans Vissers. Dr. Vissers studied for a master's in chemistry at the Free University of Amsterdam, and before completing a PhD on the modeling and packing of capillary LC columns at Eindhoven University of Technology. For the past 20 years, Hans has been working in the mass spec research facility for Waters in Manchester. So I think here we're sort of linking the Netherlands UK axis that we've already seen this morning, because you will be speaking from the Netherlands for this session. Over the past 20 years, Hans has acted as a principal scientist in life sciences and proteomics, and as a senior manager in pharmaceuticals and life sciences, before taking on a more strategic role in scientific operations. Currently, he is the senior consulting scientist for scientific operations, and in this role has engaged with a variety of groups around the world devoted to providing mass spectrometry solutions to the many scientific questions being posed by the current pandemic. With his presentation, Hans aims to inform us on the experiences from mass spectrometry based projects for SARS-CoV-2 detection, the application of mass spectrometry in COVID-19 research. So over to you, Hans.
Thank you very much, Kevin. So you should be able to see my screen.
Yep.
It shows up. Okay, excellent. So as the title was already mentioned, I will skip the first slide in the interest of time. So I'm going to try to talk about the application of two types of mass spectrometers for two different application areas, actually. So the first one is MS for diagnosis, so which has been addressed during the first two talks. And that will be the application of a so-called tandem or triple quadrupole mass spectrometer. And that type of instrument reads out nominal mass, basically, so with a accuracy of 1 Dalton. But the benefit of the instrument, however, is it's fast, it's sensitive, and it not only measures the precursor mass of a molecule, but also its product ion. So in that regard, it brings in specificity, because you measure two pieces of mass spec information, and you often combine it with the separation technique as well. So there's three pieces of technology going on over there that provide specificity. And the other application is the application of mass spectrometry for more rapid detection, so direct analysis. And here, a high resolution type of mass spectrometer is typically applied, and those type of instruments provide accuracies of a few millidaltons. So in terms of specificity from a mass resolution perspective, those instruments are more specific than the tandem quadrupole mass spectrometers, which affords direct analysis. So I will try to provide examples of both during the next 20 to 25 minutes. I guess the first question you could really pose is why you would like to apply LC-MS for these type of studies. The first two talks addressed the analysis of SARS-CoV-2 by reading out the genetic information. The benefit of LC-MS would be that you read out the protein information directly. So it's an orthogonal quantitative readout of proteins by measuring, in the case of the tandem quadrupole mass spectrometer, of digested peptides. The other benefit is that many clinical laboratories are already equipped with those type of instruments, so translating the method could be relatively easy. And I think the third advantage shown over here is an attractive one from a analytical development perspective, because if a protein mutates, it's often a single amino acid substitution, which means from a mass spectrometry perspective that you only have to dial in a different mass that you would like to monitor. So once you have developed a method, if a mutation of the virus occurs, being able to detect that and quantify that mutation should be relatively straightforward. It's also believed that the method is less prone to contamination compared to real-time PCR-based assays, because you don't amplify. Basically, you directly read out the protein content of the virus. And thirdly and lastly, we think that this is key, actually. You can you can multiplex. So the mass spectrometer does not know what it needs to detect, it just records mass. So in potential, you should be able to detect and quantify multiple viruses or multiple proteins if you would like to do so within the same experiment. So what makes mass spectrometry an alternative attractive compared to PCR type of techniques? The main reason is the protein content of these type of viruses compared to other viruses. So the NCAP protein was already mentioned, as well as the spike protein during the first talk. I'm overviewing here their main roles. So compared to other viruses, the number of protein copies, if you want to call it like that, per viral particle is very high. So in the case of the NCAP protein, it's believed, it depends on which manuscript you read, that there are around 300,000 protein copies per virus particle. And I'm highlighting a little table over here in the bottom. It's actually also from Erasmus Medical Center, from the group of Jeroen Demmers. He did a culture study and he tried to determine how much protein was actually present. And you can see in the table over here that NCAP, spike are together responsible for around 90% of the protein content of the virus particles. This was realized by proteomics researchers actually quite early during the pandemic outbreak. And to my best knowledge, these are the first two papers that discuss the application of LCMS for the detection of SARS-CoV-2. There's a group in Brazil, that's the paper with the dark blue header over here, that did swab analysis, and there was a group in Germany who looked at the detection of SARS-CoV-2 in cargo solution, actually. This is just a reminder in case you're less familiar with MRM, multiple reaction monitoring type of mass spectrometry. So the principle relies on the following. You have a whole cell proteome. In this case, it's an extract from a swab. You digest the content of the swab with a protease like trypsin. This generates peptides, and the peptides are being detected by the mass spectrometer. In targeted proteomics, what you do is you measure precursor mass, so that's the intact mass of the peptide. You fragment it, and then the second mass analyzer records the fragment ion. So the beauty of peptides is that you can almost predict which fragments you're going to detect, because the amino acid bond is the weakest bond within the peptide. So you can simply calculate if the charge resides on the N terminus or the C terminus, and that gives rise to so-called Y and B ions. So what you do is you dial in the mass spectrometer, say, okay, I expect this peptide with this precursor mass, giving rise to those fragment ion masses, and those I would like to detect. There are, however, some challenges associated with this as well. The first one is matrix effect, and I'll try to address that later on. And the matrix effects are actually sample dependent, so it depends on if you are dealing with swabs, with gargle solution, or with saliva. So you might end up with specific protocols for particular samples. So that's what I'm trying to highlight over here. That's less likely that a single working protocol can be implemented across laboratories. And it's also a serial process. That's something that has to be taken in mind. So an LCMS injection from run to run, it depends on the experiment type, can be anywhere between 2 to 10 minutes. And the sample preparation and automation can become relatively complex, dependent on how the sample is being prepared. Well, one of those early papers I showed was trying to address it. I guess this is actually a different paper from the Mayo Clinic, where they were suggesting to utilize robotics for this, because although trypsin digestion is relatively complex, if you are working in a clinical laboratory, I guess, and if you consider enrichment, it becomes even more complex. But they're repetitive automated steps. So robotics could help here, actually, and the benefit of robotics would be that you can prepare a lot of samples in parallel, which are then being fed into a mass spectrometer. So you could imagine cases where one automation robotic system is feeding into multiple mass spectrometers to be able to provide the throughput that is required for these type of testing. So following those two papers, a initiative was started at the University of Ghent. I believe Bart van Heuvel is actually on the call, because I noticed his name. He's one of the students involved in this, and together with Martin Stanis, they started work. And what they realized is that there are a few things being involved if you have the ambition to move these type of assays into the clinic. And so they tried to divide that up into three sections. It's the detection of specific peptide biomarkers, which is the blue section over here. Then trying to assure that you can roll out a method across different laboratories. And that's where the beauty of this consortium actually is. They reached out to the community and said, we need help with this, because we are discovery proteomics people. We are less experienced with translating this into an LCMS-based assay. Could you help us? So they reached out to academics, vendors, and also CROs actually. And I believe in total, it was somewhere between, I think it was around 15 laboratories that have contributed in total to that. And the outcome of that experiment is being evaluated now. It's published in this paper over here. That could act as a template actually to provide a method that could be utilized in clinical laboratories. And I don't know if the audience is aware of the moonshot program going on in the UK. There, a group of academics is trying to develop this method further actually. So I think the main output of this work and publication has been the input for a larger program being executed in the UK at the moment, trying to implement part of the results discovered during this experiment into a more high throughput assay. But what I'm going to talk about a little bit on the next few slides is the sample preparation and related techniques, because I think that's a bit more complex compared to gene readout. So what the group did again first is they created some digestion experiments, and they realized quickly, and that's the case in Belgium, that the majority of the samples being provided to laboratories are actually from swabs, and I believe that was mentioned during the first talk as well. In the case of the Belgium swab samples, they are typically in universal transport. Excuse me. So what Bart and Martin were focusing on is developing a fast and robust digestion protocol that would allow you to detect, or firstly digest those two proteins we have been talking about earlier, spike and ANCAP, in the presence of that particular matrix. And that comes with challenges of its own. But what they realized, and after talking with one of the members of the consortium, that especially the ANCAP protein can be digested relatively quickly. So a 15-minute protocol was developed that allows digestion of the protocol, which generates a relatively not large number of peptides. So this was still done as part of the discovery experiment. So a typical proteomics experiment was conducted to detect them, but at least a semi-quantitative experiment was conducted to see if there was an increase in signal response when the amount of recombinant protein, in this case, was increased as well. So this formed the basis of the selection of the peptides, in combination with some bioinformatics experiments where the group of Leonard Martens was looking at which peptides were conserved actually well across different species, I believe, mainly in variants of the protein. Once that was achieved, the group reached out to the consortium. So the experiment as a whole, if you look at it, it's relatively straightforward. So it's a protein present in a universal transport medium. Then some sample processing is being conducted. In the case of the Ghent, excuse me, in the case of the Ghent experiment, the protein is precipitated in acetone and is directly digested. Cleanup in this case was not considered because it will make the protocol more complex. So the resulting solution was directly injected on the column. But if needed, an additional sample cleanup step could be included over here. And then the MRM analysis was being conducted, showing that schematic over here again, how the instrument operates in these type of analysis. I'm highlighting this over here because at this stage of development, one still has the option to target both target and non-target peptides, if you want to call it about that. So you can target both the virus and anything else. So what I'm not going to talk about, I will just highlight on one of the next few slides, is you could also try to detect host proteins. So that will tell you something about the sampling efficiency during the early stages, during swab collection of the proteins. And that can be used to normalize, for example, the sample. So when this was being tested, it was also realized that to be able to advocate LCMS as an appropriate technique, correlation with PCR is probably required. So a number of samples, and I believe this was from Leuven in Belgium, clinical samples, 20 patient samples were analyzed by both PCR and by LCMS. And what's plotted over here is the CT value obtained by PCR, and the area on the curve is measured by mass spectrometry. So this is simply the sum of all the peptides and the fragment ions that are being detected. And what you see over here is pairwise identification of the proteins or the virus particle of PCR. So for instance, if I highlight this one and this one over here, they correspond, and that one over here. So you see a nice trend line. However, what you also see is that PCR, in this case, was able to detect to roughly 30 CT equivalent values, whereas with LCMS, the ability to detect something roughly was limited to CT equivalents of around 20 to 21. We know that the mass spectrometer is more sensitive because if we analyze the recombinant protein in solution, we are able to reach CT equivalents of around 25, which brings us closer to the required detection limits, I assume, and that you would like to utilize in the clinic. So the question is why that is. Well, the main reason, excuse me, is because of the presence of matrix. So UTM matrix, or universal transport medium, is containing albumin and collagen, and those are digested as well, and they hamper the detection. I was talking about the pairwise, oops, the back one, pairwise display of the proteins. So this plot over here tries to correlate PCR with mass spectrometry. So here you have CT values here on the curve of those pairs. You see a fairly linear correlation between the two methods. Should mention over here those three red markers. Those were patient samples that were negative, and those were not detected by mass spectrometry. So in this particular small data set, there was good agreement between the discrimination between positive and negative samples between the two techniques. So I was talking about the matrix. I guess you need to have a little bit of mass spectrometry background, that I guess to understand what this is trying to show. This is solvent, so that there are no compounds being detected in this region of the chromatogram by the mass spectrometer when just clean solvent is being injected. If the same area is being summed in a blank comprising UTM matrix, this is what you see. So that's what's hampering this experiment. The other thing that the Kent group looked into is the importance of consumables for these type of assays, and that's listed over here. And they do have an effect actually on the peptide response readout that you are able to achieve. So here are just a few of the consumable types being mentioned, but collectively you can see if you make clever use of certain consumables, you can increase signal or lower limits of detection by roughly 50%. The other thing that was optimized during this stage was the digestion protocol, was reduced to 50 minutes by talking to one of the consortium members. However, as you see on the plot over here, it does affect the peptide readout. For certain ones it does, for other ones it doesn't. And if you look at this particular well-responding peptide, digestion protocol and matrix type or swab type didn't have a great effect, but for others, it was completely reversed. So there's something to bear in mind when translating this method into the clinic, that ideally it should be as robust as possible, because I guess the thing you do not want is that when there's a shortage, for instance, of UTM swabs and you have to switch over to e-swabs, that it will have an effect on quantitative readout. Well, speaking about quantitative readout and standards, I guess that's the main topic of this webinar, is the need of standards. And in my mind, it depends a little bit on the workflow type. So if it's very targeted, if you know which peptides from which proteins excuse me, you would like to target, you could synthesize stable isotope-labeled peptides. The drawback of that is that you will only be able to detect and quantify those standards, those peptides quantitatively. The alternative would be, and that's being advocated in the paper, is to synthesize a concatemer. And the abbreviation for that would be a Q-concat. And that's an artificial protein that's being synthesized that comprises peptides for different proteins of interest. So in this particular Q-concat, there's 2 spike peptides and a number of NCAP peptides, but also some histone peptides. I was talking about the sampling efficiency earlier. So by including those, you could co-detect those together with the virus particles, and you can either normalize on it or use it as information to try to assess sampling efficiency. And these 2 peptides, or those few peptides over here, that allow you to normalize retention time if retention time variation would occur. But I guess it's a balance between 2 things. So if the method is very targeted, this might be a very viable alternative. But if more, and probably it will give you the optimal sensitivity, especially if you could enrich those peptides. But if you're still in the stage where you do not know which peptides would work best, a Q-concat might be a better alternative. So now trying to translate this back to where we are with LCMS. So this is a paper we found and was being used as a management tool to discharge SARS-CoV-2 patients. And if we try to annotate that plot with someone who we call infectious, easily detected with LCMS and PCR, then that person or patient would reside in this range. This is probably the more clinical critical detection range by either PCR or LCMS, and the previous speakers have touched upon that before. And when you have a CT value readout of 36, that patient is often qualified as non-infectious anymore by means of PCR and can be discharged. So ideally, this is the area where we would like to be with LCMS. And the previous plots show that we're roughly over here in solution, but with certain matrix types, we end up over here actually. And this is just an additional piece of information highlighting that same area again, but the SARS-CoV-2 virus proteins being detected in different matrices, including urine, nasal swabs, blood, and so on. So one of the things the Kent group has tried to do, and we've done those calculations ourselves as well, is try to correlate CT values to actually the LCMS readout, and that's being shown over here, because what that tells you is at what concentration level you roughly would like to be in terms of the standards that you spike into your sample, so you have a balanced readout of both your native peptide that you would like to detect and the internal standard. And it's roughly over here from 90 to 25. This is that same blue area again, where we would like to be if we want to make LCMS a more viable detection technique in the clinic for the detection of those peptides. And this is just showing some example chromatograms of the readout of a mass spectrometer. For, in this case, the blue is the internal standard, the red is the native peptide. And this demonstrates proof of concept by extracting information from a standard that with enrichment, you should be able to move into this blue area. So I guess the conclusion is that some possible some enrichment strategies are required. And I've listed a few over here. So different groups are looking at all of them. So you could try to remove the virals via a decoy protein-based approach. The two that are more effectively being pursued are protein affinity enrichment and peptide amino enrichment. So both are enriching either at the protein or the peptide level. So you need either a protein antibody or a peptide antibody. And then there are groups working on FMR-based solutions. So that's a theoretical construct mimicking a small portion of the protein antibody to capture the protein. And I think this one is an interesting as well. It's a group in the UK is looking at the application of lipid removal agents to see if by removing the lipid complement, in this case from plasma, they can enrich the samples. There are some pros and cons, as in most cases with the analytical techniques, and LCMS is not an exception, is some of those, especially those protein affinity-based enrichment techniques, they require the virus and the protein still being in their active states. So if during the sampling stage the virus is deactivated, those enrichment techniques might not be applicable anymore. So the question is preservation really required for enrichment? Then the other thing you need to start talking about and thinking about is automation of robotics, because the number of steps being included into the protocol will increase substantially. And that leads then to the question, and I think Corinne addressed that over here, that there is some danger in that as well, that you might not reach the required detection limits anymore. Could we consider pooling strategies? The benefit would be that there's a possible cost and time reduction here, but the negative might be that we might not detect positive patients anymore. So I mentioned the Moonshot program in the UK. So some labs, it's 6 academic labs in total, took this on, and they were tasked by the UK government to see if LCMS is a viable alternative to PCR testing if you want to do mass population testing. I'm just highlighting two examples over here, what those groups are working on. I don't think they reached final conclusions yet, but this is just an example of one of the groups. It is University College in London. It's Winnie Haywood and Kevin Mills. They are trying to optimize the digestion protocol further. So they are looking at different denaturing agents and cleanup, and also comparing on-swap and off-swap digestion. And this is just an example where the result of an on-swap digestion was being conducted. And it seems that for one of from the proteins we're talking about before, that's more beneficial than in solution digestion. So after precipitation of the protein and then digesting it. One of the other groups is looking at enrichment. This is from the group of Don Jones together with Dan Lane from the University of Leicester. This is an example of peptide enrichment. I don't know if you can see that over here. This is a software package called Skyline. So they're still doing dilution experiments and looking at enrichment efficiencies, but at least this result shows that the enrichment is very selective. So here you have a traffic light type of system, green dots and red dots. So what the group in Leicester is trying to do or demonstrate, I guess that's what I should say, is how selective this method is. So they are targeting different peptides from different proteins, but only for the two, they only detect the two for which they have the antibody raised against. So this particular peptide over here, so you see the green dots, and that particular peptide over here. So it demonstrates at least the potential that this affords additional selectivity. So you can get background removal, and noise will be reduced, sample complexity prior to introduction of the peptide digest or digest peptides to the mass spectrometry is greatly simplified. So that should improve sensitivity, especially when you're going to combine it with enrichment. So the next few slides, I would like to spend some time on an alternative technology. So it's less quantitative. It's a more qualitative technique, qualifying patients, if you want to call it like that. And that's using a technique called desorption matrix ionization. So in the previous method, the column effluent from an LC separation was being introduced in the mass spectrometer, and the way you do that is what's actually shown over here. You're applying a potential to that column outlet. It's called a spray tip in this case, and that nebulizes the solvent into gas droplet, they get charged and being introduced. The alternative over here is that you spray a solvent directly onto a surface. That generates primary charged droplets, leading to secondary charged droplets, which are being introduced in the mass spectrometer over here. So there's no prior separation to the detection of the analytes, and it makes this a very fast technique. So here the analysis time is around 20 seconds. And the other beauty is that it can be performed under ambient conditions. So here's an example. This is a prototype instrument. This is a synthetic swab. It's made of plastic. Someone was sampled, and then with that sprayer over here, solvent is directly sprayed on the surface, and the charged ions are being introduced into the mass spectrometer. So what DESI predominantly detects are lipid type of analyte ions. So this is a typical mass spectrum from the analysis of a swab. And then there are specific regions where specific lipid classes reside. So these were negative ions, so depending on the polarity being applied to this capillary, certain classes of molecules can be well detected. But the big difference is that we are detecting lipids over here versus proteins/peptides in the previous slide. And this is an example of a paper that was recently published from the group of Salter and Takat, where he demonstrated that you can use that to classify samples by using statistical analysis techniques combined with machine learning type of approaches. But this is just binary readout, where it's showing the masses of a given lipid, and by looking at patient samples, you can see that certain lipids are more abundant in negative patient samples compared to positive samples, and the other way around. And so this provides you a profile, if you want to call it like that, which you can use to build a classification model. And it's being shown over here. All that information on the spectra can be loaded into a statistical package tool or custom tools that you have developed yourself. And in this case, a principal component analysis was used to demonstrate the demonstrated discriminating power of mass spectrometry in combination with statistical tools. Oops, that's done too quickly. And if you then try to extract from that information the corresponding mass spectrometer, mass spectra, you see the discriminating masses from the given lipids that were being detected. So these are the three principal components being shown over here, and that their corresponding features in terms of M/Z values of the lipids of interest. That was taken on board, and it's still being investigated if this could be the information used to develop a mass spectrometry screening based approach. So this is a prototype that you see over here. So this is a dry swab. So the swab is being used to collect a sample, and this is a robotic loading system. I don't have an animation over here. It's basically a tray. You put it in, and then it's being introduced into the source region of the mass spectrometer, and the voltages are being applied and the liquid sprayed onto the solution. What you need for this is actually positive and negative samples for data analysis and to develop a classification system. And in this case, machine learning was used. Other people use least discriminated analysis, PCA. I think it depends on your flavor or your interest. I think they can both be applied. But ideally, when we want to move this into clinical laboratories, we don't want to use statistical packages like that. We want to develop something more user-friendly. And what you see over here is like an iPad type of console, where the laboratory technician would place the swab onto a holder, push a button is being introduced in the instrument, and the analysis being conducted. So here are 3 or 4 screenshots. So the first one is showing start analysis, then the data are being acquired and analyzed, and then a green or a red button should appear to indicate if the patient was positive or negative in this particular case. With that, I would like to summarize. I think it's fair to say that LCMS is being explored as a diagnostic test for SARS-CoV-2 by a number of groups. And I think we're at the stage where it still needs to be decided which sample preparation method is the most robust, actually, across different matrices. But if I look at the results and after talking with those groups, I think consensus is being reached at the moment. What are the most robust peptides for detection of those particular proteins, and subsequently which standards then have to be developed to make those methods more mature, I guess. But the bottleneck is still enrichment, and that's the matrix dependency of the technique. But if the appropriate enrichment methods do become available, it will allow researchers to reach the detection limits that are required, but also increase method robustness, because then you start taking away that matrix dependency. Then the application of mass spectrometry for rapid detection and research stage is, I believe, still in research stage of development. So it's not only DESI that people are looking into. I use that as an example, but MALDI, it's similar to DESI in that you analyze from a surface. It's different in that it's being conducted under vacuum, and you use a laser to generate ions, but the principle is the same. You generate ions from a surface. Is being explored. That technique requires cleanup because it's very matrix sensitive. So that's still in research phase. And then something called acoustic mist ionization. So that's ejecting a droplet from a solution directly into a mass spectrometer being investigated as well. Here, dependent on the technique, also enrichment is probably required. Certainly for MALDI, most likely also for acoustic mist ionization. DESI, I think we're still at the stages to determine if it's required or not. With that, I would like to conclude. Thank you for your attention, and I would like to acknowledge the collaborators that are listed over here on this particular slide.
Thank you. And that brings us to the end of our presentations. Could I invite all panelists to switch on their videos, and we'll move to the question session, and I'd invite Jim Huggett and Gavin O'Connor to lead us in the question session. Thank you.
Thank you, Robert. Can you hear me now? Okay, excellent. I just switch over to a different technology. Thank you. That was a great presentation. Gavin, do you have any, given that we're following Hans straight off, do you have any questions you want to relay for the mass spectrometry?
Sure. There was one that's come in from Gustavo at BIPM. Which reference materials would be most useful for LCMS method standardization? Do you think the intact spike protein or nucleocapsid, or do you think that the peptide standards are good enough?
I think if you go down the peptide enrichment technique, I think a stable isotope is appropriate, because then it becomes a very targeted method, and you're trying to target just one or two peptides. And then that's probably the most quantitative in terms of accuracy, because you know what you synthesize, you know the quantity, but also in terms of costs, because you compare synthesizing a stable isotope label peptide compared to QConcat, there's a substantial price difference. Yeah. So it depends, but I think it's still to be decided what the most appropriate method is.
Okay, thank you. We've got about 8 minutes left, so I've got a question. There's a few questions coming, but I'd like to try and pose a question that applies to all of you. We just heard about measuring essentially peptides. Corinne spoke about antigens, and Maria about RNA. These methods are being compared across the world with different methods, with the RNA for better or for worse being used, I think, as a measure for which everything else is compared to. Obviously, the RNA may be too sensitive clinically in terms of infectiousness, but my question is more linked to the analytical component of that, and the fact that whether the challenges with comparing the various methods. Firstly, with respect to the fact that the RNA may be there and the protein not, for perfectly normal reasons that are fine for the disease, and also the RNA may not be there, but the protein is. So there's that aspect of it. And then the variation associated with the RNA measurement, particularly, we know from Professor Zeikart's presentation at the previous one of these that a given cycle threshold CT measurement, which everybody had mentioned in their presentations, can easily vary by 10 cycles or more. And so this is going to also impact on this problem. So I guess the question is to what extent are the current methods and the comparison of the current methods okay, and to what extent are they potentially causing problems? Could I ask perhaps Maria to first give her thoughts on that question?
Yeah, so thanks very much. Well, you can see through the kind of quality assessments and comparison of performance that there is quite a range of sensitivity in tests that are detecting RNA. Specificity itself seems to be very good. I think all of the tests that are in current use would be improved in their interpretation by actually having widely available, very reliable quantitative standards. And I don't think that's something that's easily enough available, particularly at the time when you are developing tests. This is not a stable situation. There is massive expansion of different types of tests and also complexity of tests, because things have started out being multiple uniplex detections. They're moving towards multiplex detections and now combining with different virus detections to try and give a syndromic approach. And I think all of these would be benefited by having reliable, quantitated RNA standards to allow much better interoperability and also comparison between laboratories. So let me stop there, and I'm sure I'll...
Thank you. That's really good. And I guess as has already been mentioned, NIBSC's work is getting and you mentioned actually that the international unit will hopefully come to that. But I'd also like to plug the work that CCQM has been doing over the last year on reference measurement procedures for RNA measurement and also antibodies as well, which will hopefully allow, in the next time this happens, for such standards to be produced at a much faster way that may be able to plug the gap should that occur. Corinne, what's your thoughts with regards to comparing the antigen testing with the RNA? Is it a fair comparison? Sorry, yep.
Corinne, you're on mute.
Sorry. Yeah, well, as I already have shown, I think it's been a choice which has been made in the meantime, because we do not have an appropriate standard to really first look into the correlation of this standard with infectivity or viability of a virus. So in the meantime, it's a comparison which is being made, but I'm not sure that it's the best we have at this moment. I mean, I think that's why.
Okay. One other question that's come up in the thing, with regards to robustness, a lot of the tests, a lot of the publications show, and this is for molecular as well, and all the others, that that it works with this performance, sensitivity, specificity. To what extent do you feel the community is, I'm not picking on the RDT, but we're talking about them, are cognizant of the fact that taking the same sample and measuring that 10 times is essentially that robustness measure? Do you feel that is being sufficiently covered? Are they working well in that context?
No, I also saw the question about the differences in lots of antigens. And no, I do not think that at this moment at this moment people are really focusing on a comparison with PCR and the clinical sensitivity, I think, of the assays, and the robustness. We really lack data, I think, at this moment to really see.
Okay, thank you very much. Gavin, please.
Yeah, this comparison that we're doing at the moment. So we've asked for RNA standards, and I'd like to ask sort of Corinne and Hans, how much of a hindrance is it? How much of a problem is it that we're comparing your technologies to PCR? Yeah, and everything is set up for PCR. So Hans, you mentioned that the transfer media and the swabs are not ideal for mass spec. you're comparing numbers of moles of protein versus CT values. Yeah. So I'd like to ask is, what is it that's important to measure? Are CT values important to measure, or do we really want to know the quantity of virions present in a specific sample?
So I guess we want to determine ideally viral load somehow, regardless of the technique. So mass spectrometry would afford you to read that out directly, but it doesn't take away the sample hindrance issue. And I think once you have a solution that you can introduce in the mass spectrometer, the readout is stable, but anything up front introduces the variation to a very large extent. So the sampling efficiency, the digestion, the extraction. So the sooner you can put a standard into the whole process, the better. And ideally that, so yeah, either a peptide or a protein that you co-digest. And if that would also be a measure that allows us to correlate with CT values, and I understand your question, Kevin, it's like, do I want to do it? No, but the community is asking for it, isn't it? So how does your test compare? Because that's being seen as the gold standard. So I guess if you could develop something that would allow us to do that, then it would become more easy to translate results, I guess, between methods. Yeah. It's not to say that one is better than the other, but at least you can say, okay, compared to this, yeah. I guess the other question that I have in these type of cases is, how accurate do you have to be during early stages of a pandemic outbreak, and how quantitative you have to be? Is speed more important than accuracy? And I guess that's something that Corine is looking at, because that's why you considering rapid testing.
Yeah, so of course for the rapid testing, speed is an important issue, but I think we should not only focus on this part of the testing, and I still think that for all the assays it would be good to have a better idea about how it correlates to infectivity. So for the mass spectrometry as well, you would want to know, I think, how exactly do the people react in their infectious phase, and when exactly are the people infectious. I mean, this clinical data would be very important to take along, I think, within the validation of the assays and setting up your cutoffs and your levels of detection that you want to have for the assay. So I think there has to be some collaboration there between the people looking into infectivity and clinical settings. Yeah, and of course the people working on the specificities of the performance of the tests. I
have a question for Corine. Thinking about the infectivity assays, could you comment more on the challenges for cell standards, developing a cell standard in terms of the source of the cells, the clonality of the cells, or the sort of spatial format of such a standard? And I think this also leads to sort of IVD testing, things like that.
Mm-hmm. Yeah, at this moment,
that's a challenge for the cell group.
Yeah. At this moment, there's not very much of standardization of the virus cultures between the labs, but within labs, of course, there has been standardization based on internal PCR standards and quantitation of assays. But I agree that there are huge steps to make there, and that would be really helpful once so that we can really internationally compare the results much better than the same effort is being made for the molecular assays, I think, at this moment.
So going back to Hans, Gavin hinted at what might work better for a mass spec-based assay. So can you comment on what types of samples could be appropriate? Would a dried blood spot ever be an option? Things like that?
The short answer is yes, but probably we need more enrichment because the concentration will be much lower. we have seen evidence that the swap solution has a great impact, because one of the things they are looking at during the UK Moonshot program is deactivating at the stage of sample collection. So rather than using universal transport medium, they use a 70% ethanol 1 molar ammonium carbonate solution, which is much more mass spec friendly than a protein digest. So the detection levels that those groups are reaching are lower compared to the initial work being conducted by the Ghent group. But the Ghent group was restricted by the usage of certain swaps within the clinic. So I guess as an end user, if you want to call it like that, you don't often have the luxury to say you have to use these swap types, because otherwise our method doesn't work. So yeah, I agree with you. It's, if you can address the sampling issue, that would help detection. But then, yeah, ideally we want to detect them in plasma and then dry blood spots. And we have seen evidence, because I mentioned the group of Leicester, that it's not the NCAP protein, but the spike protein can be detected in plasma. So there is that potential, but it requires development time, I guess.
I think there are more questions, but I'm sorry that I know a number of our speakers are going to have to leave very shortly. So I think it's an ideal moment to thank you all very much for your input, for the time you've taken. I think it's been highly interesting and allowed us to learn a lot more than we knew at the start. Thank you very much. And we look forward to organising a third webinar in some months' time. Thank you for your time.
Thanks. Bye.
Bye-bye.
Bye.
Thank you.